Good morning, everyone. Sir, can you press the hide option on the screen? Hide. Can you press the hide option, which is like, yeah. Yes, click, yeah. Go back to PowerPoint. Click on the PowerPoint. Can you, yeah, perfect. Click on this. So I welcome all. Good morning. Today we have the pleasure of having Dr. Devendra Kumar with us, our Assistant Professor, College of Biotechnology, Devasu Mathura. And the topic chosen for deliberation today is hydroponics, technological intervention into forage production and research. So you are audible as well as visible, and your presentation is also visible. Please start the session.
Good morning, sir.
Yeah, Dr. Pratika Pal.
Good morning, sir. Am I audible?
Yes.
Yes, madam, you are audible.
Very good morning to all of you. I'm Dr. Pratiksha Panda, working as Assistant Professor in the Department of Veterinary and Animal Husbandry Extension in Duvasu, Mathura. I welcome you all for today's session on hydroponics, and which will be delivered by Dr. Devendra Kumar. He is working as Assistant Professor in Department of Plant Biotechnology in College of Biotechnology, Duvasu, Mathura. Dr. Devendra Kumar completed his PhD in Agricultural Biotechnology in 2020 from Sardar Vallabhbhai Patel University of Agriculture and Technology, Meerut. He began his professional journey in August 2019 at JRF at the Division of Genetics, IARI, New Delhi. Later, he joined Mangalayatan University, Aligarh as an assistant professor in the Department of Biotechnology and Life Science in November 2020. In March 2020, he transitioned to Swami Vivekanand Subharti University, Meerut as an assistant professor in Department of Biotechnology, Faculty of Science. Since February 2024, he has been serving as assistant professor at College of Biotechnology, Tuvasu, Mathura. With more than 5 years of teaching and research experience, Dr. Kumar has developed expertise in the area of crop improvement for abiotic stress tolerance, hydroponics, plant tissue culture, gene expression, and plant breeding. He has supervised one master's students as major as advisor and has been a member of advisory committee for more than 10 MSc and PhD students. Dr. Kumar is a prolific researcher and has published 2 books, 6 book chapters, 14 research papers, 3 review articles, 5 lab manuals, and 9 popular articles. He has also successfully completed 2 institutionally funded research projects and has 3 utility patents to his credit. As a manuscript reviewer, he has contributed to various national and international journals of repute. His contributions has earned him several accolades, including Young Scientist Award in the year 2021 from Pragatishil Krishak Vikas Seva Sansthan, INSC Research Excellence Award 2021, Young Achiever Award 2019 from Agricultural Technology Development Society, and Subharti Young Achiever Award 2023. He has also delivered guest lectures and conducted hands-on training programs for both academic institutions and professional organizations. His contributions to the field of agricultural biotechnology reflect his commitments to research, education, and advancement of science. Dr. Kumar, I welcome you for today's session. I hope you enlighten all of us by sharing your knowledge and insights. Dr. Kumar, you can start your presentation.
Okay. Thank you, ma'am, for my introduction. So let me start with my presentation. So good morning, distinguished guests, respected members, and dear participants. It's my pleasure to be present the topic on hydroponics, that is the technological intervention into forage production and research. And I'm very privileged to have you stand with you all in this winter school program that is jointly organized by NADCL and Dovasu Mathura. So this is the collaboration that bring together the experts as well as researchers and the dedicated members who are working in the agricultural sectors as well as the forage production system. So this is the uh, this is the a place where we can explore our ideas as well as the research which is going on in this area for the improvement in the forage production as well as the sustainable agriculture development. So, without delaying further, I'm going to start my presentation on hydroponics, that is the technological intervention into forage production and research. So, before going uh, to the problems about the fodder production system, so we just have to uh, view on the hydroponics. Actually, what is hydroponics? This is a new technique. This is continuously going to be in practices from a later long time. But what are the advancements that has been going on, and what is going on in the research in case of utilizing this hydroponic tool for improvement of crop production as well as other crops such as the fodder production. So hydroponics is nothing new. This the term that is comes from the Latin language that means working water. Hydro that means water and polo that means labor. So we can say this is a soil-less growing system. Soil-less growing system that because we all know there is nothing nowhere is mentioned about the soil is necessary thing. The soil is necessary key for growing any type of plant or any type of crop. So the necessary thing that is required to grow any crop, that is the mineral nutrient supply, water as well, and other is the light. So being together or taking together with all aspects, the hydroponic technology was developed that can provide the precise control over the environment conditions and as well as for optimizing the all parameters for the optimal plant growth. So this is a technique that have the nutrient rich aqueous solution that has emerged the transformative technology in the forage production and research by offering the precise control over the factors such as water, nutrients, temperature, light, pH level, and other platforms such as relative humidity. All parameters can be controlled in a area that is laboratory that is called as a hydroponic laboratory. This tool can provide us with the high throughput phenotyping technique that for the rapid crop evaluation uh, characterization of the genotypes at early stage, that is the seedling stage. So this system allows to screen the large population within a single time. So it provides us an opportunity to characterize the genotypes as well as the uh, rapid production of the crop system. So this technique can be considered as a reliable tool as well as the efficacy to for the repeatability of the crop trials in the field area also. So I can say this would be a technique for the future farming or urban farming as well as the forage production as well as in the research. So how this technique can be considered or can be included in the forage production research. So uh, just before that, we are going to towards to know the different types of hydroponic system at this time that are available in the market or that are available and developed by using the different technique, that is static solution method. In this, the nutrient solution is always static and the nutrient supply is provided through the different channels to the plant for growing, and different type of method one another, that is the continuous flow solution method. In this, what happens, the nutrient supply is continuously provided by a flowing system or by using a pump. So this solution is going to be continuously flow and provide the nutrient supply to the plant. Another one is the aeroponic method. In this method, the nutrient supply is provided by using aeropon. Air means the roots of the plants are kept into the air, and one of the parameter is this you define, that is the time, and there will be a spray of at the time different time interval to the roots that can provide the nutrient supply to the root system for the crop growth and production. Another method is the fogoponics method. In this fog-like structure, the fogs are created by using different type of nozzle supply of the nutrients that can provide the nutrient supply to the roots of the plants, and the plant is going to be rapidly grow. And another method of the hydroponic system, another type, that is the passive sub irrigation method, such like of uh, different type of supports are used for growing the crops, and the sub-passive irrigation of hydroponic solution is being to be provided for the proper growth and rapid growth. And another is the rotary method, and sub and flow sub irrigation method. And one of the most important part of this presentation, which I'm going to be highlight, that is the deep water method that is being to be used in case of hydroponic research system. And this is uh, this technique, yeah, this tools of deep water method is going to be utilized for the initial screening of the genotypes for early stage characterization. So the hydroponic mechanism talking about, the mechanism is consists of different system. That is, first one is the growing medium. In this growing medium, almost all the things that is required to support the plant growth and the plant system is utilized. And another thing, that the growing medium that have the nutrient solutions, and this supports the plant roots. And for supporting the plant roots, different type of perlite mixture, vermiculite mixture, or coconut coir, even just water can be utilized. And in nutrient solution, there is a balanced solution of essential nutrient that is required for the optimal plant growth, that is the macronutrients, micronutrients, iron supplement, and other plant growth regulator. Whatever is required is supplemented in the form of nutrient solution to this system. And then there is another thing, that is the water delivery system. Water delivery system is provided in case of, if there is if we are using this type of system, then what the water is provided by adding the nutrient solution in this. But in case of, if we are using such like of system, such as continuous flow solution method, static solution method, so there, or fogoponic method, or aeroponic method, then there should be a water delivery system. The water or nutrient solution is delivered to the plant roots by using different methods, such as growing pumps and another things. So you can see in this figure clearly that step one, that is consists, that showing one of nutrient solution container that provided to the system, and another that is the drilled hole into the container lid. Actually, this system is depicting about the deep water method. In this, the container, one or another type of container is used, and will be a lid that have the different type of drilling system. And this drilling system, there will be a container lid or such type of container lid, that is the drilled hole into the system, or any other system mixture is utilized, such as cotton, perlite mixture, vermiculite, coconut coir, and other things that can support the plant. And then another step, that have the assemble of air pump into the container. Air pump actually uh, provides the air supply to the water for the particular respiration to the system, and another one that there will be a reserve water into the container and nutrient as well as the adjust pH level. pH actually have the significant role in this hydroponic system because the plant have the specific pH requirement for translocating its nutrient, and then there will be the assembling of the hydroponic system that have the container as well as the lid and the attach the plant into this container lids. Fourth one, that is the water reservoir. You can see here this black colored line is showing about the water reservoir that have the nutrient with water supply into this, and there is a net pot that is providing a support to the plant for growing, and there will be styrofoam or vermiculite mixture that supporting the plant weight, and there is a tubing, one of the tubing system that is provided with the air pump that provide the air supply by using the air exchange, air stone. Growing tray or growing channels, another component where the plants are placed, and then those are allow the nutrient solution to flow over the roots and facilitating the nutrient uptake. And the last one that I already told about, that the air pump and air stone, actually these are used to supply the oxygen to the nutrient solution, proper aeration that prevent the root rot and promoting the healthy growth. Because aeration is necessary to the plant, so the oxygenate, yeah, aeration supply is compulsory. Uh, so this system having the limitations to this, in case of agar in what happens if the supply of the air is prevented or there is disturbed. So in that case, what happens? The plant growth is not going to be the sufficient, and it will go to the lesser production. Now, talking about what are the production constraints in case of fodder production or any type of crop growth or crop production, or for the sustainable supply of the fodder. So we all know about there is a round year supply of the fodder is required for the animal growth as well as the production or productivity. So in case of crops, what happens actually with the productivity loss? Uh, Actually, this is the main thing which disturb the sustainable supply of the fodder production is these days is the climate change as well as the soil that influences the plant fitness. As we all know, the climate changing scenario is going to be changed, and these climatic factors all directly affect the physiological as well as the different parameters of the plant growth, and ultimately it disturbs the homeostasis of the cell. These climatic factors include the atmospheric gases, light, temperature, humidity, precipitation, as well as wind, and these climatic factors may vary seasonally. According to the season, these parameters are going to be changed, and climate projections suggest that this time these are going to be changed. Uh, uh, and we all know that the IPCC also suggests the temperature is going to be increased in upcoming in future time. Also, this time the mean temperature also has been increased, and uh, if we see the climate change projections of this year, we can say this year was a very high temperature, and the photo period in the polar regions change during the year from the continuous light to the constant darkness. And another thing, first one is the changing climate scenario, and another thing that going to be a hamper the plant production or production being a production constraint, that is the abayotic factor. Abiotic factors as well as biotic factors always have a direct link or direct connection with the plant growth as well as the production, because abiotic factors influence each other. Uh, for example, if there will be a highlight, then there will be increase in the air temperature, or if there will be increase in the air temperature, it will modulate the evaporative cooling, transpiration, increase transpiration rate, that will directly affect the plant production. And we can see ocean currents also regulate the atmospheric temperature as well as the rainfall patterns. Another thing that is the edaphic factor. Edaphic factors include such as soil mineral, organic composition, ion exchange capacity, pH, microfauna, microflora, as well as the climate, that can determine the availability of air, water, nutrients into the soil to the plant. Because the soil porosity depends upon the particle size, so there if there will be the uh, high particle size, higher particle size, then there will be the aeration will be greater in the highly porous soil. So these all factors being together is going to be hamper the plant productions. And for optimizing these all factors, we require a high throughput phenotyping tools as well as such type of system that can modulate or that can optimize these things for the sustainable production of crops or fodder to the round year. And these imbalance into these all factors have the primary as well as the secondary effects on the plant. For example, if there will be the reduced water potential, then and cellular dehydration, that will directly alter the physiobiochemical properties of the cells, that will lead to the secondary effect on the plant growth, reduced metabolic activity as well as the production of ROS, that will also lead to the cell death. So these are all factors that hamper the crop production or continuous growth of the plant. So this time, what we need actually, this is the need of the day. We require such type of systems that can sustain our crop production and that can provide the round year supply of the green fodder to the animals, as well as the such type of systems are required that can provide us with the elite genotypes. And what are the solutions for these actually? Actually, the solution of these, we require the crop for new season. What will happen actually? The climatic change pattern, yeah, climatic patterns has suggested us the temperature is going to be increased, or there will be rainfall patterns are changed in different seasons, and variability in these parameters has had emerged as a requirement of the crops for new seasons. And crops with the alternate maturity is required, because we can see or we all know okay, at the time of maturity of the crops, there will be emergence of different types of insects and pests and different type of diseases as per the seasonal variation, changing in the temperature as well as changing in the relative humidity. The requirement is the crops with the alternate maturity, so that crop is going to mature within the changing climatic changing scenario. Before that, changing to the parameters into changing into that variables of temperature as well as the emergence of new insects in further season. If we have the crops with alternate maturity or previous maturity crops, we can escape our crops with these type of problems. And another thing that require this time, that is the crop that can sustain year-round production. Year-round production, because in case of livestock sector, we can say the continuous green fodder supply or continuous fodder supply, that can provide the nutrient supply to the animals for their growth and production is required. So sustaining the around year production is required. Crops with the better tolerance to abiotic and biotic stresses, because the major factor that loss to any type of crop production, that is the abiotic factors and biotic factors, actually these simultaneously act, and this will lead to the production loss of up to 70% if there will be when proper management are not being done. So we require another option for this. We should have the crops with better tolerance to these abiotic stresses. Another thing, that is the more diversity is required in the crops, because fodder crops uh, are required for around year production. So we need diversity in the crops with the better tolerance ability, with the better nutrient enrichment or nutrient nutritional content, as well as crops with new spatial arrangement. So we can provide the continuous supply of the fodder to the animals, and crop with the higher nutritional content to secure the nutritional security to animal health. And last one, that is the new improved germplasms are required, and this area is required always more and more diversity for any type of crop improvement program. There will be always requirement is that is the more and more diversity, so we can improve the newer germplasm with the better traits, with better nutritional content, as well as better production rates with better tolerance ability. So, how the hydroponic can help us into the forage production research for developing such type of new germplasm crops with the better tolerance, with the abiotic and biotic stresses, and crops with more diversity, crops with higher nutritional content. These can be solved by the hydroponics. Uh, how? So let's see. Hydroponics is the noble step towards the crop improvement research. How actually this can provide the control over the environmental variables like nutrient level, water availability, and pH and temperature, etc. And such as light parameters can also be optimized, light parameters can also be controlled. Actually, hydroponics can provide us with the uh, control system or controlled environment conditions. So we can optimize our conditions to according to the crop requirement, as well as our objectives for we are going to be work in case of if we are going to do the work of in the crop improvement research. So crop improvement research, actually what required, there will always be there is requirement of the high throughput phenotyping tools, and hydroponics can be a high throughput phenotyping tools. Actually this can provide us with the phenotyping at the time of seedling stage, and it can provide us with the early characterization of crops or different type of genotypes. It can enable the evaluation of large population within a lesser area. You can see here in this system, I already described about there are different types of holes are drilled into the lid of the container that can provide. We can arrange our different genotypes, yeah, different germplasm of a single variety genotype, whatever we are working for improve that particular crop. We can arrange them according to our trials, and we can utilize the system into the forage research studies. Key traits like biomass production, stress tolerance ability, and quality. Actually, this can provide the uniform controlled conditions for consistent results and accelerate the identification of elite genotype by providing the uniform solution. Uh, because in case of soil, if we are going to analyze, as we are going to assess any type of crops tolerance regarding its abiotic stress, any type of nutritional content, any type of availability, bioavailability, anything else. Suppose let's take an example of abiotic stress. If we are going to assess our genotype for several type of abiotic stresses, for example, salinity stress, that is the major stress in case of crop production, uh, so in that case, we can assess a large population by using hydroponic system in a lesser area in a single plate. We can assess more than 100 genotypes and 200 genotypes and 50 genotypes. That will be dependent on our models of the system that we had developed according to our uh, experimental design. So this can provide us with the larger population assessment within a single time, and we can have the opportunity to optimize the different parameters such as different type of EC control, different type of drought stress treatment, as well as different type of nutritional supply, effect of different nutrients can also be optimized. So, in case of we are going for the screening for stress tolerance, actually hydroponic system can be useful technique, yeah, high throughput phenotyping tools for assessing the genotype, yeah, plant germplasm or genetic mutant for identifying the traits that are associated with the stress tolerance. This approach can accelerate the breeding program that are aimed to develop the stress tolerant forage varieties. In the system, you are seeing here the different genotypes are can be grown by using different germination papers, and then these genotypes can be planted into this system, and then assessment for the specific type of stress tolerance can be carried out by using this hydroponic system. This can be utilized in case of if we are going to do the nutrient studies for, suppose we are going to utilize, you're going to assess different type of different genotypes of different crops for nutrient uptake. So researcher is able to investigate how plants can absorb or utilize the nutrients from these solutions, and it can study the different effects of the nutrient concentration as well as different combinations on plant growth as well as plant development by assessing its morphological traits, physiological traits, biochemical traits, as well as the molecular traits. The nutrient deficiency studies also can be conducted by utilizing this hydroponic technique that can help in understanding how this nutrient deficiency can impact the plant growth and how the plant can adopt to this such type of deficiency. Hydroponic tool can be utilized in case of for a research for studying the root architecture, because for crop improvement the root provide key important parameters for any type of stress tolerance ability. So studying the root characteristics is a requirement for the breeding program. So root architecture can be studied by utilizing this hydroponic tools. This can provide us with the easy access to the root morphology, density of root, branching patterns, as well as different type of growth patterns and interaction with the nutrient solution, how this nutrient solution or how this roots or plant can interact with the nutrient solutions, and it can provide a critical understanding for the nutrient water as well as the uptake of the solution, and it can provide water or nutrient with the and we can optimize actually we can actually optimize the different parameters for different thing. All these characters at a single platform, we can say hydroponics can be a tool for the early stage characterization, and it can make possible to analyze the germplasm at seedling stage by uh, morphological characterization such as root and fruit analysis, physiological and biochemical characterization, different type of biological, physiological as well as the biochemical parameters can be assessed by utilizing this hydroponic system such as water content, water deficit, relative turbidity, chlorophyll content, protein content, and other different type of enzyme assay can be conducted by and utilizing this hydroponic techniques as the early stage characterization tool. And these different type of characterization or different type of parameters such as morphological, physiological, and biochemical parameters can provide the understanding to the researcher and breeder for improving the forage crop performance. And it can help to identify the traits that are linked for the higher yield and better quality. Apart to this, it can provide us with the identification of elite germplasm or any type of fodder crop. Because in case of plant crop production, there should be the quality production is dependent on different availability of quality seeds or quality germplasm. Because if we have the quality germplasm, it will definitely produce the more production. So it can help us with the identification of elite germplasm by acid, and identification of elite germplasm can accelerate the breeding programs, and it can identify with the crops with having the higher resilience to different type of abiotic stresses and having the different desirable traits. For example, if we are going to uh, going to improve any forage crop, so there will be requirement of the crop with the higher biomass, with high vegetative growth. So by utilizing this system at can provide us the opportunity at the seedling level, and it can provide us with early characterization of the germplasm for at the seedling stage for with the desirable traits, and the elite germplasm can be further utilized for producing the crop with higher production. And this system can be utilized for further productions also directly by because it can be integrated with the advanced technologies, for example, by combining the genomics, imaging techniques, as well as the automation techniques. It can increase the precision as well as the efficiency in the research as well as the further production, green fodder production around the year, and it can improve the data collection as well as the large scale screening. You can see here different companies are utilizing this hydroponic technique for producing the green fodder around the year by using different types of this system. Now, talking about the practical benefits of forage production, actually, it can enable us with the year-round cultivation, with the higher resource efficiency. It can be ideal platform for the area which have the limited arable land and where the climatic conditions are very harsh and rigid at the time, and it at there in such type of places we can produce with the high quality forage for the livestock feed, as well as the different practical benefit is it can provide us with the faster growth. Actually, the hydroponic system works automatically and by utilizing the automation tools in this hydroponic techniques can provide us with the complete nutrient mixture as well as water with the roots growing in the plants, as well as plants get everything whatever they need at the time, at the real time, whatever they need is provided by this system, and they do not waste a lot of roots for searching for any type of nutrients. So plant growth is going to be faster in case of utilizing this hydroponic tools. And as we are growing these crops with a controlled environment and within the laboratory form, so it can provide us with no weed or pest emergence. So if there will be no emergence of the weeds and pests, there will be the problem of elimination of weeds can be minimized, and there will be no requirement of the spray for the pesticide as well as insecticide, because mostly insects and pests are breed or live in the soil, and we are not utilizing the soil in this system, so we don't need to use the any type of pesticides or toxic chemicals. So we will produce the powder with the no pesticide residue, no pesticide content, or no toxic chemicals. So at last, at the conclusion, I can say that hydroponics can be a revolutionizing tool for forage research as well as forage production, and it can facilitate the rapid crop evaluation and trait improvement actually, and it can enhance the sustainable livestock feeding system. It can pave the way for more resilient and high yielding forage crops. Thank you. If there is any question, kindly.
Okay, if anyone have any query, please ask to the Dr. Devendra.
हेलो नमस्ते सर।
Namaste, Srinath Bahul.
हाँ सर, आई एम दिनेश आचार्य फ्रॉम राजूवास। सर एक प्रश्न था कि हाइड्रोपोनिक जब फॉडर प्रोडक्शन अपन करते हैं
Yes,
तो
yes,
इसमें ना कुछ सीड जर्मिनेट नहीं हो पाते। सर आवाज आ रही है?
yes, yes, yes,
हाँ सर, जो हम सीड यूज करते हैं फॉडर प्रोडक्शन के अंदर हाइड्रोपोनिक मशीन में,
yes, yes.
तो कई बार जर्मिनेशन कम होने के कारण वो फंगस डेवलप हो जाती है सर।
सर इन दैट केस एक्चुअली रिक्वायरमेंट क्या होती है कि हमें जो सीड्स जो हम यूज करते हैं फॉर फॉडर प्रोडक्शन,
जी
उनको
सर।
एक बार जो है स्टेराइल करने की जरूरत पड़ती है। उनको एक स्टेराइल की ट्रीटमेंट जो आप यूज कर सकते हैं, सच एज कोई भी माइक्रोबियल या एंटी फंगल एजेंट्स आप यूज कर सकते हैं फॉर द ट्रीटमेंट ऑफ दैट पर्टिकुलर सीड। जैसे कि कोई भी एंटी फंगल एजेंट, जैसे कि चाहे वो कार्बामेट क्लास का हो, जैसे
जी
कॉमन कार्बेंडाजिम हो गया, मैंकोजेब हो गया, या कोई भी एंटी माइक्रोबियल सप्लीमेंट जैसे कि हम सोडियम हाइपोक्लोराइड का ट्रीटमेंट दे सकते हैं। और उसके ट्रीटमेंट के बाद अगर आप जो सोइंग करेंगे तो
जी।
जर्मिनेशन बहुत अच्छा होगा, और उसके अलावा कोई भी किसी भी तरह के कंटामिनेशन का जो प्रॉब्लम जो है वो सॉल्व हो जाएगा।
लेकिन सर अगर इसको ऑर्गेनिक वे में हमें लेना हो तो क्या करना चाहिए सर?
सर एक्चुअली जो हाइड्रोपोनिक टूल्स जो है इनकी, तो अगर प्रोडक्शन की हम बात करें कि फॉरेज प्रोडक्शन के केसेस में
जी सर।
हम अभी कुछ लिमिटेड ही क्रॉप्स हैं जिनमें हम इसे यूटिलाइज कर पा रहे हैं। अभी इन
जी
पर जो फर्दर रिसर्च की रिक्वायरमेंट है कि इसको एज अ वर्टिकल फार्मिंग सिस्टम के रूप में डेवलप किया जा सके, और अधिक से अधिक और भी क्रॉप्स हैं जो कि इनको ग्रो किया जा सके, यूटिलाइज किया जा
जी
सके। और एक तरीके से मैं कहूँगा कि ये जो टूल जो है ये एज कम्पेयर टू प्रोडक्शन से ज़्यादा उनके रिसर्च में ज़्यादा अच्छे से यूटिलाइज किया जा सकता है। जैसे कि मैंने अभी फोकस किया था कि हम इस सिस्टम को यूटिलाइज कर सकते हैं फॉर द आइडेंटिफिकेशन ऑफ एलिट जीनोटाइप। अगर हमारे पास एलिट जीनोटाइप होंगे जिनके अंदर जो कैरेक्टरिस्टिक्स अच्छे हों, क्वालिटी अच्छी हो, न्यूट्रिशनल कंटेंट अच्छा हो, उस तरीके के अगर आइडेंटिफाई करते हैं, क्योंकि किसी भी तरह की क्रॉप के प्रोडक्शन के लिए सबसे पहली जो प्रिलिमिनरी रिक्वायरमेंट है वो है क्वालिटी सीड।
जी सर।
तो ये जो है क्वालिटी सीड्स और क्वालिटी जो जीनोटाइप जिनके पास डिजायरेबल ट्रेट्स है उनको आइडेंटिफाई करने में हेल्प कर सकता है ये सिस्टम। उसके अलावा फॉरेज प्रोडक्शन में तो डिफरेंट कंपनीज जो इंडस्ट्री जो है वो यूटिलाइज कर ही रही है, क्योंकि इंडस्ट्रीज का काम जो होता है वो तो बेनिफिट के लिए होता है, वो तो हमेशा जो उन्हें पैसा कमा के दे उस चीज पे काम करते हैं।
हाँ सर, इसमें क्या है, अगर क्वालिटी जो क्वालिटी सीड लेंगे ना सर, लेकिन इसका कॉस्ट ऑफ कल्टीवेशन बढ़ जाता है सर। तो जो
जी जी
इजीली अवेलेबल जो सीड होते हैं सर मंडी के अंदर,
जी।
वो ही हम लेके आते हैं और उसमें फिर ये दिक्कत आती है सर।
सर उसके लिए सबसे पहला तो तरीका यही है कि आप जो है सीड्स को ट्रीट करके जो सोइंग करेंगे तो आपका
जी
जो एटलीस्ट मैं कहूँगा कि मोर देन 70% प्रॉब्लम तो सॉल्व ऐसे ही हो जाएंगे। क्योंकि आपके और भी केसेस में देखिए, जैसे कि सॉइल के केस में आप देखिए, अगर सॉइल में आप डायरेक्टली जो सोइंग करते हो, बुवाई करते हैं आप किसी भी तरह किसी भी सीड्स की, तो
जी सर।
अगर सीड अगर ट्रीटेड नहीं है, सीड्स को अगर प्रॉपर ट्रीटमेंट नहीं मिलता है तो सॉइल बोर्न डिजीजेस भी होती हैं। और इनके केसेस में कहीं ना कहीं अगर हम ये कह रहे हैं, जैसे अगर हाइड्रोपोनिक को यूटिलाइज कर रहे हैं तो इनिशियल कॉस्ट ज्यादा हो रही है।
जी सर।
लेकिन अगर हम इसी चीज को जो सीड्स हम इसके केसेस में यूज कर रहे हैं, अगर उन्हें हम सॉइल में डायरेक्टली सोइंग कर रहे हैं विदाउट ट्रीटमेंट, तो वहां पर सॉइल बोर्न डिजीजेस के केसेस ज्यादा होंगे। तो किसी
जी
चीज
सर।
का अगर सर कहीं पे बेनिफिट है तो कुछ जो डिमेरिट्स भी होती हैं। तो अभी क्योंकि ये डेवलपिंग स्टेज में है, इन केस ऑफ अगर प्रोडक्शन की बात करें डायरेक्टली फॉर फॉरेज प्रोडक्शन या फॉडर प्रोडक्शन, तो
जी
ये
सर।
अभी सिस्टम को और डेवलप करने की जरूरत है, इसको और रिसर्च की रिक्वायरमेंट है कि ताकि हम वर्टिकल फार्मिंग सिस्टम की तरफ जा सकें। और इस केसेस में हम कह सकते हैं कि क्योंकि आने वाले समय में जब जो है, क्योंकि एरेबल लैंड जो है या कल्टीवेबल जो लैंड जो है वो लैंड की अवेलेबिलिटी कम होगा, क्योंकि जिस तरीके से जो अर्बनाइजेशन होता जा रहा है या बढ़ता जा रहा है,
जी
तो
सर।
उस तरीके से लैंड की रिसोर्सेज की अवेलेबिलिटी कम होंगी, तो ये जो है अर्बन फार्मिंग के रूप में कंसेप्ट जो है डेवलप होगा।
जी सर।
और आपका जो क्वेश्चन है सर, उसके लिए सबसे बेटर यही है कि आप जो सीड्स जो आप यूज करते हैं उनको एक बार आप ट्रीट करके बिजाई करेंगे, उनकी सोइंग करेंगे, तो वो समस्या जो है सॉल्व हो जाएगी।
ठीक सर, थैंक यू सर, टॉपिक बहुत अच्छा था
और
और हम राजस्थान में हाइड्रोपोनिक पे बहुत जोर दे रहे हैं सर।
जी 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 सर, इसके अलावा एक सबसे बड़ा, क्योंकि हाइड्रोपोनिक जो है हमें ऐसी अपॉर्चुनिटी जो प्रोवाइड कराता है कि हमें क्योंकि कंट्रोल्ड हमारे पास में जितना भी जो एनवायरमेंटल जो पैरामीटर्स हैं, जैसे कि टेम्परेचर हो गया, लाइट हो गया, रिलेटिव ह्यूमिडिटी, पीएच, ये सारी चीजें जो है क्या कि वो हम ऑप्टिमाइज कर सकते हैं अपने तरीके से। जैसे जिस तरीके से मान लो सपोज अगर हम कोई ऐसी क्रॉप ग्रो करा रहे हैं जो कि जिस समय पे उसका कोई सीजन नहीं है फिलहाल में, तो उस तरह की क्रॉप को भी इसमें ग्रो कराया जा सकता है। लेकिन उसके केसेस में इस सिस्टम को थोड़ा सा और मॉडिफाई करने की जरूरत है। सच लाइक जैसे अगर हमें टेम्परेचर जो, जैसे इस समय विंटर की क्रॉप है, विंटर की जो विंटर सीजन जो चल रहा है, तो इस समय की जो रबी क्रॉप की जो सीजन की जो क्रॉप है वो ग्रो कराई जा सकती है। लेकिन यूटिलाइजिंग हाइड्रोपोनिक्स we can grow the other crops also by providing the different या setting the hydroponic tools in a such type of laboratory या such type of cabin जहाँ पे हम टेम्परेचर को रेगुलेट कर सकते हो, तो हम उनको भी ग्रो करा सकते हैं। तो इसकी ये मेरिट्स भी हैं। तो ये जो है एक तरीके से एक प्लेटफॉर्म हो सकता है ईयर राउंड ग्रीन फॉडर प्रोवाइड कराने का।
जी सर, थैंक यू
और
सर।
जी जी जी, उसके अलावा रिसर्च में तो यूटिलाइज किया ही जा सकता है फॉर द डेवलपमेंट ऑफ द क्रॉप जीनोटाइप विद द बेटर टॉलरेंस एबिलिटी, क्योंकि बेटर टॉलरेंस अब हमारे पास जो जिन जीनोटाइप्स, अगर हमारे पास टॉलरेंट जीनोटाइप होंगे किसी पर्टिकुलर स्ट्रेस के लिए या पर्टिकुलर किसी डिजीज के लिए, तो वो
जी सर।
डिजीज की समस्या ऑटोमेटिकली सॉल्व हो जाएगी।
जी सर। जी सर।
और डाइवर्सिटी तो रिक्वायर्ड है किसी भी तरह के जो क्रॉप को इम्प्रूवमेंट या क्रॉप इम्प्रूवमेंट प्रोग्राम जितने भी होते हैं, उन सब के लिए ज्यादा से ज्यादा जितना डाइवर्स जीनोटाइप हमारे पास अवेलेबल होगा या जितनी जीनोटाइप्स की हमारे पास अवेलेबिलिटी होगी उतना बेटर होता है। और इन केस ऑफ अगर हम फॉर्डर के केसेस में देखें तो वहां पे हमारे पास जो जीनोटाइप्स जो है वो लिमिटेड है अभी। तो इस टूल को उस पर्टिकुलर फॉरेज रिसर्च एरिया में भी यूटिलाइज किया जा सकता है। वहां अगर हम ज्यादा फोकस करें तो हम क्वालिटी सीड्स या क्वालिटी जीनोटाइप प्रोड्यूस कर सकते हैं सर।
ओके ओके सर।
जी
जी सर, थैंक यू सर।
वेलकम सर।
हाँ जी।
And if anyone not agree, but this training, this session we will be in Dr. Devendra for a very nice session and providing detailed insight about the hydroponics and because.
Sir, actually your voice is going to be break in between. I think the इंटरनेट कनेक्शन का कुछ इश्यू है।
हेलो। हाँ डॉक्टर साहब। ओके ओके डॉक्टर साहब, थैंक यू।
यस
थैंक how are you? Thank you for joining and your I'm not able to just overlook your presentation, but reflects very good, and definitely this will be helpful for the participants also. Thanks on the behalf of NADCL and from Duvasu Mathura. You are also the part of Duvasu. So thank you, Dr. Saab, for joining.
so thank you for all for providing me this opportunity, and in conclusion I can say that hydroponics will be a powerful technology that can reshape our agriculture system as well as the forage production system, and it can provide us with studying the complex traits as well as it can utilize a platform or a tool for early stage characterization of crops with higher tolerance ability. So thank you so much, sir, for
Thank
providing
you,
me
Dr.
this
Saab.
opportunity. So
Thank
let
you.
me finish this. And can I?
Okay. Thank you, Dr. Saab. Thank you. For participant, we can now have the meeting after 2:30 p.m.